Assalamualaikum and good afternoon. I Bitusa, Mujib and all my fellow friends. So today we're going to present chapter 6, which is the vision and visual perception. As for myself, Nur Maisara binti Suhaidi, I will present the first subtopic of the chapter, which is light and visual apparatus. There's 3 parts into this subtopic and we're going to dive one by one. So let's jump right into the first part, which is the visible spectrum. So before I start, what is the stimulus for vision? I think most of you guys know what's the stimulus for vision. I even put a clue there. Yes, it is light. Today we're going to talk a little bit more about that. So visible light is the part of electromagnetic spectrum. Yes, let's go into physics. So electromagnetic spectrum includes a variety of energy forms, from the highest frequency, which is gamma rays, down to the radio waves, which has the lowest frequency. So the visible part of the electromagnetic spectrum only accounts for 1/70 of the frequency range. Most of the frequency range are not even useful for producing images, such as radio waves, for example. It's not useful for producing images except radar because it passes right through objects. But some, like X-rays and radar, it can actually produce image, but it requires powerful energy resources and special equipment. Other than that, heat-producing objects can also give off infrared energies, and some animals, like the nocturnal animals, actually use this energy to detect prey at night because we couldn't see at night. So the heat that is produced from the living things can actually give off infrared energy. Humans can convert those energy into image, but we need to use machines. However, infrared are only best for detecting nearby objects. As for the further objects, distant objects, it will produce more blurred and fuzzy details. So our sensory equipment, our eyes, is adapted for detecting energy that is most useful for us, which is the visible light spectrum. So the visible light can actually produce well-defined image because it is reflected from the objects with minimal distortion. It is a form of oscillating energy and it travels in waves, just like sound energy. We can actually describe visible light in frequency, but then the numbers would be too large, so we normally describe it in terms of its wavelength. Wavelength is the distance oscillating energy travels in one complete cycle, and the unit is in nanometer. And different wavelengths actually produce different colors of visible light. For example, let's take a look at the diagram at the right side. So the light ranging from 500 to 570 nanometer that strikes our receptors will actually produce the color green. So it really depends on the wavelength. So we are done with the stimulus. Let's go to the organ that is responsible in accepting the stimulus. Okay, the second part is the eye and its receptor. So the eye is a spherically shaped structure and it's filled with a clear, thick solution that is called aqueous solution. And this sclera is opaque except for cornea, which is transparent because it receives the light. So behind the cornea is the iris. Iris is actually what gives your eyes its color. And iris is a circular muscle that controls the amount of light. It contracts reflectively in bright light and it relaxes in dim light. So other than that, pupil is a hole and it's actually black because the light doesn't come out of it. So behind the iris is the lens. This lens is a transparent but a very flexible tissue. If you can see at the second diagram, there's a muscle attached to each end of the lens, and it is responsible to contract and relax when it wants to focus on a certain object. So when we see a far away object, the lens will be flattened by the muscle and vice versa. So that's how the light rays can be focused on the retina. Alright, let's focus more on the retina. Okay, retina is a light sensitive tissue at the back of the eye. It is made up of two main types of light-sensitive photoreceptors called rods and cones. We will discuss more about them in a bit. So the light must pass through the neural cells to reach the photoreceptors. So the photoreceptors, like this is the sclera, this is your eyes, this is your sclera, and this is the photoreceptors. It is connected to the bipolar cells first, the green one, and then the bipolar cells is connected to the ganglion cell, the orange brown one. So this ganglion cell will connect directly to the optic nerve. So photoreceptors are filled with light sensitive chemicals called photopigments. Photopigments absorbs energy from light and it breaks down molecules. Let's talk mechanisms. So how does the eye detect light? Alright, so these two rods and cones are responsible for detecting the light. These are actually photoreceptors. So let's look at these two components and differentiate the roles between these two. So the rod pigment is called rhodopsin and the cone pigment is called iodopsin. The difference between these two is that rod absorbs light much faster than cones, so which is why it is best for low light situation, because in daylight there's light all the time, right? So it will be broken down most of the time, while cone, it requires a high light intensity level, so it functions best at daylight because that's where we got most of the light. And then the second difference is rod only has one type of rhodopsin, which is why it is only for monochromatic vision, and cone actually it has three types of iodopsin, which is the red, green, and blue one, and it's responsible for the color vision. As for the distribution of the cones and rods is actually distributed through the retina. It's in the peripheral area and not at the center of fovea. Fovea is the tiny part in the middle of the retina, and it has high visual acuity. This is why it will produce good detailed vision, because the cone is concentrated at the center of fovea. Other than that, cone actually has one ganglion cell for each cone. Meanwhile, the rods, they have to share a few ganglion cells. That's why it has poor detailed vision. And other than that, a cone has large receptive field, which contribute to the light sensitivity. Receptive field is the area of the retina which a ganglion cell receives its input. That's why it is more sensitive to light, because it's distributed around the peripheral. So there's so many parts which has the cones, while the cone has small receptive field. The cone system has small receptive field because, like I said, the cones are mostly distributed only at the fovea, so it has small receptive field. So let's move on to the last part, which is the pathways to the brain. Like how does the light move the information of the stimulus from the retina down to the back of the brain. When the light enters the eye, it will actually go to either two pathways, which is the blind spot or the optic nerve. So let's see the blind spot first. So the blind spot, as you can see at the diagram there, it is where the nerve exits the eye, because as you see at the blind spot, there's no connected optic nerve there, right? So there's no receptors, which is why it is called blind spot. So the blind spot of the two eyes fall at different points in the visual scene, so you won't notice any part of your visual world missing. And also your brain is actually good at filling all the missing info from the blind spot. And then the second pathway is to the optic nerve. Let's look at the optic nerves. This is where it gets tricky. These two optic nerves, each from every eye, will run to a point just in front of the pituitary. So as I speak, you guys must focus on the diagram, okay? If not, you guys will not understand the pathway, okay? Okay, and then they join at the short distance at the optic chiasm, and then they will separate again and travel to the first synapse at the lateral geniculate nucleus. And then at optic chiasms, the axon from the nasal side of the eye cross to the other side and go to the occipital lobe in the opposite atmosphere. So where is this nasal side? Nasal side is here. Okay, this is your nose, nasal area, and this is the nasal side of your eyeball. Okay, and then the neurons from temporal side, which is the outside side do not cross over, but they will go to the same brain. So remember, this is the nasal side and this is the temporal side. Okay, it's quite confusing, so let's move to the next slide. I will break down each process one by one so we will understand together. Alright, so how the splitting output of the eye does not cause distortion of image? Before I start, do not mix up between the right retina and the right visual field. So let's take a look first at A. A is at the left visual field, and then A from the left visual field falls on the right half of each retina. The right half, okay, both of them, right half here. And then the B from the right visual field falls on the left half of each retina here. See the cross? You see? Okay, and then the second step, they will move all the way down to the optic chiasm, and this is where it will cross path. The A from the left eye cross with the B from the right eye. So the blue part will go to the right side and the red part will go to the right side. So as you can see, the blue from the left will cross with the red from the right, so both will come together with their respective matching colors. And then the information end up that the information from right half of the retina will be transmitted to the right atmosphere. I'm sorry. Okay, last part of this subtopic is the retinal disparity and depth perception. Depth perception. Okay, so how can we see the world in 3D? There's actually a reason why our two eyes has some distance. This distance is responsible for our retinal disparity. So retinal disparity is the difference in location of an object's image on our two retinas. So when we look at something, either it's far or near, our eyeballs will move, right? So this movement of our eyeball will actually create an angle from the distance of the object that we're looking into. So this angle is called convergence angle. So the smaller the angle of convergence, the further the object we see. So this information from the angle and the distance, the retinal disparity, will be detected in visual cortex, and then the different neurons fire depending on the amount of the lateral displacement, basically the distance between the displacement. So the information is passed down to the anterior parietal cortex to be combined with other information such as the object shape and location. So that's how we can perceive distance between different objects. So that's all from me. Thank you.
Hi guys, so my name is Nuaira Fatini binti Khairul Saleh, and I'm going to present about my part, which is color vision. So what is color vision? Okay, the color vision is the ability to differentiate or to distinguish the various wavelength of light and to perceive the differences, such as the differences of the color tone. Okay, so the color vision is used to determine the location, the shapes of objects, the identity, and the characteristics. Okay, so there are three theories that related to the color vision. So first is the trichromatic theory or Young-Helmholtz theory. Okay, the second one is opponent theory, and the third one is combined theory. Okay, let's discuss about the first theory of the color vision, which is trichromatic theory or Young-Helmholtz theory. So this theory is proposed by Thomas Young and Hermann von Helmholtz. First of all, Thomas Young proposed that the development of the color vision is come from the actions of three different receptors. And he suggested that the eye contained the different photoreceptor cells that sensitive towards the different wavelength of lights in the visible spectrum. After that, another researcher, which is Hermann von Helmholtz, he expanded upon the Young's original theory. Okay, so Helmholtz suggested that the cone receptors of the eye have three conditions of wavelength. First, short wavelength, blue, medium wavelength, green, long wavelength, red. He also proposed that how our brain interpret the color from the environment was influenced by the strength of the signals detected by photoreceptors. Helmholtz also discovered that the people with the normal color vision need three wavelengths of light in order to create the different colors through a series of experiments. Hence, Young and Helmholtz proposed a trichromatic theory, which is just three receptors in the retina that are responsible for the perception of the color. Okay, so one receptor is sensitive to the color green, another receptor is sensitive to the color blue, and other receptor is sensitive to the color red. So the combinations of these three colors will produce the colors that we are capable to perceive. So the photoreceptors tend to have different sensitivity levels. So the blue receptors are the most sensitive, and the red is the least sensitive. And then the ability to perceive the color, it requires the interaction between at least two photoreceptors. But if the three colors of photoreceptors combine together, it will form any visible color in the spectrum. Okay, so for example, when we watch TV or look at any electronic device, we can see the application of the trichromatic theory on it. So this is because all the colors that we see on the screen are made up from the different intensities of the tiny red, green, blue dots or pixels of light. So this is illustrated in a color cube as shown at the left side of the slide. Okay, so let's go to the second theory, which is opponent theory. So this theory is proposed by Ewald Hering at 1878, and this theory is going to explain about the color vision, but in terms of opposing neural process. Okay, and this theory also suggests that our ability to perceive color is controlled by the three receptor complexes with opposing actions. So there are three receptor complexes, which are red-green complex, blue-yellow complex, and black-white complex. So these cells can only detect one color at one time because two colors oppose one another. So we cannot see the color of greenish red, yellowish blue, or the combination of black and white color because the cells can only detect one color at one time. Okay, how opponent color works? Okay, so opponent color works through a process of excitatory and inhibitory response within the receptor complex. Okay, for example, for red-green complex, the red creates a positive or excitatory response while green creates a negative or inhibitory response. As you can see there, it occurs within the receptor complex, red-green complex. Okay, so these responses are controlled by opponent neurons. So opponent neurons is the neuron that have an excitatory response towards some wavelength and have an inhibitory response to another wavelength that opponent part of the spectrum. The opponent process theory also explains the perceptual phenomena of negative after image. So negative after image is the colors that we see are inverted from the original one. So you can try several activities that can show you the negative after image. So one of it, I have put it here in this slide. So I put the rectangle there with two parts. So the first part is the white color and the second part is green color with the red dots. Okay, so the instruction is you have to keep focus on the red dots for 30 seconds to 60 seconds, and then you can switch your vision towards the another part, which is the white part, and blink for a while. Okay, and then you will see the results. So based on the theory, we can see the green color of the dots at the white part of the rectangle. So I'm glad that you can share with us your result too. after we have shared to you the slide. So I hope that you try this activity after we give to you this slide. Okay, so we go to the last theory of the color vision, which is combined theory. So since the trichromatic and opponent theory are seems to have a contradictory, then Hurwitz and Jameson resolved the conflict. So they propose that our eye have three types of color receptors, which is red, green and blue, and these color receptors are interconnected in and the opponent process at the ganglion cells. So actually the combined theory want to tell us that there are three color process at the receptor and four beyond the ganglion cells. Okay, so as you can see here, there are three colors of receptor cell and four colors of ganglion cell. Okay, so there are four conditions that happen between them. So first is long wavelength light will excite the red cones and the red-green ganglion cell. So as the result, it will give the perception of red color. Okay, the second is medium wavelength light will excite the green cones and inhibit the red-green cell. So it will reduce the firing rate at the ganglion cell and give the green color and give the signal of green color to the brain. Okay, the third is short wavelength. Okay, short wavelength light will excite blue cones and inhibit the yellow-blue ganglion cell. And as the result, it will lead to a perception of blue. So what happened to the light midway? Okay, light midway occur between the sensitivities of red and green. So because of that, it will stimulate the both cone types. So what happened after that is the firing rate in the red-green ganglion cell will remain unchanged. This is because the equal stimulation and excitation from the two cones would cancel out. Okay, however, the red-green ganglion cell will remain unchanged, but the yellow-blue ganglion cell will excite. So the cones connections to the yellow-blue ganglion cell are both excitatory. So their combined excitation will excite the ganglion cell. The ganglion cell is the blue-yellow ganglion cell, and as the result, it will produce the perception of yellow. Okay, if all three cones are stimulated at equal intensity, so the red-green ganglion cell and the yellow-blue ganglion cell, both of them receive an equal amount of excitatory and inhibitory stimulation. So as a result, it will produce a perception of a shade of grey. Okay, the theory is considered success if it is consistent with the known facts, can explain the those facts, and predict the new findings, and can predict the new findings. Okay, so the combined theory meets all of the three criteria. So the first one, because it is consistent with the observation that all the colors can be produced by the red, green and blue light. The second one is it can explain why observers regard red, green, blue and yellow as a pure colors. It also explains complementary colors, negative after effects and the impossibility of color experience such as greenish color, greenish red color. Okay, and the third one, it predict the discovery of three photopigments and of the excitatory and inhibitory neural connections at the ganglion cells. So there are a few research regarding to the combined theory. So if you want to know more about the research, so you can read it in the textbook. Okay, to conclude all the three theories, so the trichromatic theory, it occur at the retina of the eye. And the opponent theory, it occur at the ganglion cell after the retina, all the way to the brain. Okay, so the combined theory is the combination of the two theories, and yeah, it occur at the retina and after the retina. So I think that's all from me. Thank you, and I will give the next presentation to the next presenter. Okay, thank you.
Hi, my name is Situna Safrina, and I'm going to continue the subtopic under the color vision, which is color blindness. The color blindness are inaccurate term called color vision deficiency, and it is occurred when a person or individual has a poor or absent from one or more cone type. So there are a few types of the retinal color blindness, which is a red-green color blindness and a blue-yellow color blindness. A person who is a red-green colorblind will see these two colors, but unable to distinguish or identify between them. Either the protanopia with the absence of the red receptor or deuteranopia with the absence of green receptor. So this thing same with the person who is blue-yellow colorblind that will experience tritanopia with the absence of a blue receptor. So this color vision deficiency can be detected by having subject match a short color object or with the Ishihara test. Okay, this is a picture, a plate from the Ishihara test for a color blindness. Most of the people will see number 74, but a person who with color blindness will see number 21. For a person who are total colorblind cannot see any number. Okay, the picture on the right side is the example type of the color blindness that a person with the color blind will see or experience. So I'm going to proceed to the next topic, which is form vision. So these are the subtopic under the form vision. In our visual cortex contain a map of retina known as a retinotopic map, which means the adjacent retina receptor activate the adjacent cell in the visual cortex. So these are the picture of the retinotopic map. Now we go to the contrast enhancement and itch detection. Contrast enhancement mean to make a process to make image clearly, while the itch detection mean a process to identify or distinguish where is one object end and the next begin. So why these two things is important? It is because in the first step in the form vision is detecting the object boundary. In this case of boundary, our retina use lateral inhibition to facilitate the itch detection. Hemangrid illusion and Marchbank illusion is the examples of the demonstration of lateral inhibition. Okay, for Hemangrid illusion, lateral inhibition will cause you to see a small greeny blotches at the intersection of the large square. For the Marchbank illusion, as you can see here, each band is constant in brightness, but when this band meets, you will see a slight enhancement in the brightness at the edge of the lighter band and a little bit darker at the edge of darker band. So this brightness contrast effect at the edge help us actually to see the boundaries of the object. Okay, for example, you try to see a lion that camouflage in a tall dark sky is one of the how the retina use lateral inhibition. So how our retina produce illusion using the lateral inhibition? Actually, our receptors field in eye have both excitatory and inhibitory region. And this figure, as you can see here, the red arrow indicate the excitation while the gray arrow indicate inhibition. When you see a stimulus or light, all the ganglion cells are activated. The ganglion cell 7 is activated most compared to cell 1 through 6. It is because the cell 7 receive more stimulation from a brighter stimulus, but receive less inhibition from the receptor. Okay, for cell 8, it receives a minimal stimulation from the stimulus but receive more inhibition from the receptor compared to cell 9 to 15. So as a result, as you can see here, the light bar appear lighter at its border and the dark bar also appear darker at its border. Okay, now we go to the effect of light on the center and surround of the receptive field. There are two types of the ganglion cell, which is on-center ganglion cell and off-center ganglion cell. Okay, in on-center cell, the light in the center increase firing while the light in off surrounding reduce firing below the resting level. Okay, the on-center ganglion cell, it will respond if the center of receptive field is stimulated. Okay, for other ganglion cell have an off-center ganglion, off-center and on surround. Okay, for the off-center ganglion cell, they will opposite to on-center ganglion cell. Okay, what we learned on the previous slide will be applied in this slide in order to discover the effect of the border on an on-center and off-center ganglion cell. Okay, for this figure, the light falling across the entire field will have a little or no on a ganglion cell firing rate. So it means that the excitation and inhibition cancel each other out. Okay, for a second figure, the light fall only at the off surround. So when this happens, it will suppress the firing in the ganglion cell. Okay, for the third figure, the stimulus or a light fall on the on center and only a part of the off center. So it means that the ganglion cell will firing at its maximum. For first until third figure, it is on center ganglion cell. Okay, for the last figure, it is off center ganglion cell. So on this figure, it happened when the receptor are stimulated by an object that is darker than its background. So why we must know all this thing? Because the dark stimulation is also as important as the light stimulation. So now we're going to discover about a theory in form vision, which is Hubel and Wiesel theory. Hubel and Wiesel theory begin to explore the receptive field of the cortical cell by projecting a bar of light on the screen. So they found that an actively responding cell would decrease its responding when the stimulus is moved to another location or rotate in a slightly different angle. They discovered this thing after they projected a visual stimuli on a screen in front of the visual cortex of a cat. So this is the example of their experiment setup. Okay, this figure showed the change in response in one cell when the orientation of the stimulus is varied. Hubel and Wiesel called this cell as simple cell because it responds to a line or edge that is specific orientation and its specific place on the retina. So for this part, Hubel and Wiesel explained about surprising shift in specialization in the cortical cell. If you see here on the figure, the field is overlapped because the receptor share their output with the multiple ganglion cell. So when the edge is in the position, the ganglion cell for each of the circular field increase its firing. Then the ganglion cell connect to the same simple cell, which is also firing, indicating that the edge is detected. So this mechanism is not just to detect the light dark contrast, but also to detect the contrasting edge, such as an object that is lighter or darker than its background. Okay, Hubel and Wiesel found this figure as a complex cell because when the edge move horizontally, the complex cell will continue to respond even though there are a simple cell will take over. The spatial frequency theory is another theory in form vision. The spatial frequency theory means that the visual cortical cell do a Fourier frequency analysis of the luminosity variation in a scene. So this theory is important for us To recognize an object as an object, assign it color under very light condition and detect its movement. Okay, in spatial frequency theory, we have two types, which is spatial frequency and low spatial frequency. And now we go to the illustration of the high and low frequency in the visual scene. Okay, the picture on the left side shows the illustration for the Spanish artist, which is Salvador Dali. Okay, in the middle photo show the low frequency because it produces more gradual change between light and dark, while the photo on the right side show the high frequency, which is having amount of light over large area in a picture. Okay, you can get a same effect from the picture on the right side with the middle photo by looking from a distant or partially close eye. When you do this thing, you will see the face of Abraham Lincoln. Okay, for this part is about role of high and low frequency in a vision. So, the picture on your right side is original photo. Okay, on the middle photo, it also same with the original photo, but the high frequency is removed, and the photo on your right side is when the low frequency is removed. If you notice that the high frequency change in contrast show the border and fine detail, while the low frequency define the characteristic through the shadow and texture. So I think that's all from me. Thank you.
Assalamualaikum and hi, my name is Fatin Syafiqah Nur Ain binti Fauzi, and then my topic is the perception of object, color and movement. I will be going to cover the first three topic, the first three part, and then while the other four will be covered by my friend. So let's take a look. The first one is visual processing. There are two visual processing that happen here, which are modular processing, and then the other one is hierarchical processing. What is modular processing? Modular processing is segregation of the various component of processing into separate location. So it is a segregation. Meanwhile, the hierarchical processing is lower level of the nervous system, and then it will analyze the information and then pass the result to the next higher level for further analysis. Okay, next. But the neuroscientists reject the idea of modular processing because it claims that visual information is either distributed, which is occur relatively in wide area of the brain. Okay, next. There are two pathways of visual analysis, which are the first one is parvocellular system, and then the second one is magnocellular system. Parvocellular system, the characteristic are the first one, smaller ganglion cell. Meanwhile, for the magnocellular system is bigger ganglion cell. And then the parvocellular system, small amount of ganglion cell. Meanwhile, for the magnocellular system is large amount of ganglion cell. And then circular receptive cell, that small and color opponent characteristic in parvocellular system. So it shows that the characteristic here is small and color opponent. So it gives the function for parvocellular system is the discrimination of fine detail and colors. Meanwhile, for the magnocellular system, circular receptive cell that are brightness and movement characteristic. So it will give the function of magnocellular system is for the brightness, contrast, and then the movement. Okay, now let's take a look at the example of parvocellular system. Imagine that you are trying to read a newspaper at dusk and you are unable to do so because it is high resolution and color sensitivity is low. So you are unable to read it because the color sensitivity is low. And then the next one is for the movement. For magnocellular system, what you can do is the first one, you outstretch your arm while you are facing upward. You outstretch your arm, and then you move your hand while wriggling your fingers, and then when you can notice your hand, you stop. But you can barely see the fingers, but you are able to detect the movement. So you are able that your hand is moving. Okay, the next one is there is two picture of bicycle here. The first picture, we are unable to detect that that is a bicycle because there is a different background of bicycle and then the background, so we are unable to see the picture itself. Meanwhile the other one we can see the picture clearly because there is a contrast different of color between the bicycle and then the background, so we are unable to see the object that are showing the picture of the bicycle. Let's take a look at Parvos nervous system. The first one is dominate ventral stream, and then it is carry what visual processing. The pathway is V1 until V5 and then pass toward inferior temporal cortex where V1 until V5 carries the function of color perception, meanwhile inferior temporal cortex give the function of object recognition. Meanwhile in Magnus nervous system it dominates dorsal stream and then visual processing is where, and then the pathway is V1 until V5, or we known as MT, and then pass to posterior temporal cortex where V1 pass toward V2 and until V4 it carries the information of orientation, movement and retinal disparity, meanwhile V5 give the function of directional sensitivity, and then posterior temporal cortex give the function to locate an object. And these two pathways travel to lateral geniculate nucleus and then toward the primary visual cortex, which is V1. Okay, this is the pathway that I have mentioned before. V5 or MT, and then the nearby area that receive the input from the MT known as MST are sensitive to the movement. Okay, let's take a look when we are reading. So when we're reading, we move our head from left to right to read the words, but the image move across our retina is moving, but we don't see the world around us when we are reading is moving because there is a movement sensitive cell. The function is it is sensitive to movement in a particular direction, and then some reverse preferred direction of movement as head move. Okay, so what happens if there is a damage in ventral stream and dorsal stream? Ventral stream that I have mentioned before, it is related to Parvos nervous system, meanwhile dorsal stream related to Magnus nervous system. And then when you have trouble in ventral stream, you have trouble visually identifying the object because it carries the function of recognizing an object, and then you are able to work toward and around the object and reach for them. Meanwhile for dorsal stream that carry the function of movement, so you are unable to orientate gaze on object, reach accurately, shaping hand to grasp an object using visual cue because you have the trouble regarding your movement, but you can identify the object. So these two pathways then proceed toward prefrontal cortex, which is the function here is manage information stay in memory while it being used to carry out the function that depend on the two pathway. Okay, then we continue to disorder of visual perception. The term is agnosia, which is lack of knowledge. There are two agnosia, which is object agnosia and face agnosia. Object agnosia is impaired ability to recognize an object while the intellectuality is still intact. So there is a bag, for example there is a bag, and then the person with object agnosia are able to see the object and then they are able to explain the shape. It is in rectangular shape, and then they have a tire, they have a handle, but they are unable to recognize that the object they are seeing is a bag. Okay, for next for the face agnosia, or we call as prosopagnosia, it is impaired ability to visually recognize familiar faces. The characteristic is have no problem with memory, so they are able to identify the individual with other characteristics. So they are able to differentiate between people A and people B is different due to the other characteristic, for example their speech, and then the visuality acuity not impaired, so they are able to differentiate people A and people B, their emotion. For example, people A is showing emotion of angry, people B is showing emotion of sad, so they are able to differentiate the facial expression, but they are able to identify who are they. There are two areas that are responsible for face recognition, which are FFA and medial temporal lobe. FFA located in inferior temporal lobe, that are fusiform gyrus on the underside of temporal lobe, that is important in face recognition. And then prosopagnosia it shows that there is a damage in the right hemisphere, but the phase processing of the image itself involved in both hemisphere. Meanwhile, in medial temporal lobe, located in medial temporal lobe, and then its function in memory, decision making between familiar and unfamiliar faces. Okay, so the causes are two. First one is brain damage, and then the second one is heritability. So it detects defect of the prosopagnosia does not appear on FFA, but fMRI shows that FFA also respond toward the faces as in normal individual, but in fact interior temporal and frontal cortex are diminished. So it proves here that the face recognition is distribute function despite modularity of its component. Next, cortical blindness. It is loss of visual secondary to damage to the visual pathway posterior to the lateral geniculate nuclei. So they are able to track the movement, discriminate the color, identify the emotion, and avoid obstacle because they have the blind sight, which is the ability to respond to visual stimuli that are not consciously seen, because the pathway through superior colliculus is directly to extraocular area bypassing V1. And then lastly is visual word form area, VWFA. It is written to word as a whole. For example, when we see the word a book, we see the word as a book as a whole, not by word, not B-O-O-K, but we see that written word as a whole, and then we see the word and we perceive the information as a book. I think that's all from me. I pass to my friend. Thank you.
Hi, my name is Farah Nadira binti Aburaza, and I'll continue with the next agnosia. The next agnosia is color agnosia. What is color agnosia? It is the loss of the ability to perceive colors due to brain damage. Before we go further on about this agnosia, we need to understand the concept of color constancy. What is color constancy? It is the ability to recognize the so-called natural color of an object despite the illuminating wavelength. Or in other words, we can say that the brain wants to see an object as a particular color all the time, irrespective of what the lighting condition is. Can you see the picture that I have put in the slide? The three pictures in the different lighting conditions. So let me just touch about the banana. You see that banana tends to reflect more light in the yellowish region of the spectrum compared to with the other object, regardless of the spectrum of light shining on all of them, and you concluded that the banana is inherently yellow. So why color constancy is important? Because without color constancy, color would be an unreliable cue to object identity. For example, your rental car would be unrecognizable in the evening glow. Next, we move on to how is color coding different from the wavelength. In 1983, Zeki illuminated red, white, green, and blue patches with a red like each patch set off firing in V1 cells that preferred long wavelength regardless of its actual color. Cells in V1 responded only when the patch's actual color matched the cell color preference. So in conclusion, cells in V1 is where the wavelength coded. Meanwhile, cells in V4 provide color constancy. Change and Desmond in 1990 on how V4 cells provide color constancy. Okay, first thing first, when the green light falling on the center increases the cell's firing rate, and then the green light falling simultaneously in the surround reduces or eliminate the increase, and the cells subtract out the color of any general illumination. And finally, V4 cells allow us to see our friend's face as normal pink rather than as the color that it might reflect. Okay, we return back to the color agnosia. The scientific name of this defect is cortical color blindness or cerebral achromatopsia. It occurs when people have lesions between V1 and the fusiform face area where the V4 is located. However, it is uncertain whether the deficiency can be attributed to V4 malfunction. Movement agnosia. So, movement is detected by neurons in V1 and beyond. Meanwhile, area V5/MT is the place where the information is integrated. There is a case, we call it LM case, a 43-year-old woman who suffered a stroke that caused a bilateral damage in the general area of MT. The result was movement agnosia and impaired ability to detect movement. So strange, right? Okay, so what happened is she could distinguish between moving and stationary objects only in her peripheral view. She had trouble detecting the movement of people if there were more than 2 people in the room. She also often surprised to notice that an object had changed position. Besides, she also had no sense of the object traveling through the intermediate positions. For example, when she poured coffee, she could not tell that the liquid was rising in the cup, so she would keep pouring till the cup overflowed. And finally, LM's most severe impairment was inability to detect radial movement. So radial movement is important to tell us that we are approaching an object when we walk or drive, because all the environmental objects around the central point appear to move outward. This effect provides information about our heading and is important for personal navigation. Next subtopic is neglect and the role of attention in vision. Posterior parietal cortex will combine input from visual, auditory, and somatosensory area to help individual locate object in space and to orient the body or to adapt the body in the environment. And if there is a damage in the right cerebral hemisphere, impairs the ability such as reaching for object and often produces neglect, such as ignoring the left side of the plate. So neglect is due to a deficit in attention. Can you see the picture in the slide where the model shown is the perfect whole house, but when they ask the patients to draw the model, they only draw not even whole of the house, and that is what neglect here means. The fact that when the external attention is impaired, so is visual functioning. Final subtopic for this chapter is the problem of final integration. So people keep wondering where all the information about the object is brought together, and how the brain combines information from different areas into a unitary whole is known as the binding problem. Okay, let's say, imagine watching a person walking across your field of view. The person is moving, shifting orientation, and changing appearances as the lighting increases and decreases under a canopy of trees. At the same time, you are walking toward the person, but your brain copes easily with the changing size of the person's image and the apparent movement of environmental object toward you. So it seems that a central center at the end of the visual pathway would combine all the information about shape, color, texture, and movement, constantly updating your perception of this image as the same person. So what happened if there is damage in that area? What happened is it would produce symptoms that similar to blind sight, but that affect all stimuli. And to answer the first question, which is where all the information about the object is brought together, the brain task is a balancing act between combining relevant information and also separating insignificant information. We will spend more time examining on how the brain sorts out and uses information when we talk about consciousness in the next chapter. Thank you very much. That's all. Thank you for your attention.